we will continue with the regulation of gene expression. So in short, we know that gene expression is the decision by the cell, or these are the reactions performed by the cell to decide which gene to be expressed at what level. So the concentration of a protein has to be determined by the cell, and the types of proteins present in a cell at a particular point of time also has to be decided by the cell. So the decision, the taking of this decision is what we simply represent the regulation of gene expression. In prokaryotes also, even though bacteria is a single celled organism, it's a comparatively a simple system is there. There are examples, or there you can see various methods of regulation in bacteria. So most of the regulation occurs in the transcription site, as the which is the most suitable site and most economically feasible site for regulation. So these regulatory molecules usually interact with the DNA, and this causes, or this will prevent, either prevent or enhance transcription. So there will be transcriptional level regulation. Then you can see, as the transcription initiation is the major site of regulation, and there are various proteins which are supposed to bind onto the DNA near to the site called promoter site, and this will either positively affect or negatively affect the transcription. All right, so there are different proteins involved in the interaction to this promoter regions. Okay, and these proteins have been of either of three types. One is the specificity factor. So these are certain proteins which interact with specifically to the promoter site, and which alter the specificity of RNA polymerase enzyme for a promoter. And the very common example is the sigma factor. So the sigma factor will regulate which RNA polymerase, or which promoter has to be bound by an RNA polymerase, and thus transcription has to initiate. So sigma factor is a real regulatory molecule, either to transcribe or not to transcribe a gene. The second one is a repressor protein. So these repressors will block the RNA polymerase, usually binding to the promoter site. So there is a negative effect on binding by the repressor, and activators at the same time will have a positive effect on binding to the DNA. They will promote transcription. In bacterial system, as already we have seen in the previous videos, that the genes are usually clustered together, and the mRNA, usually we call them as the polycistronic mRNA. So since the bacterial system is polycistronic, the regulation is also occurring as together, right? Means that genes associated with a particular pathway or a metabolism will be clustered together, and they are regulated by a single regulatory system. Okay, so genes with related function are usually clustered together in the chromosome, and hence it will be easy to regulate them together. Okay, so such a system, such a cluster of genes which are under a single regulatory system are usually denoted as operon. Okay, so this is what we call operon. These are nothing but a number of genes will be there, and there will be a set of regulatory units also for these genes. And the speciality of these genes are that they are usually involved in a particular pathway. They are associated, and most of the bacterial genes are located in any operons, right? At the same time, in eukaryotes, operon systems are not commonly found, right? So there'll be a common promoter and regulatory site for this operon, and the regulation occurs as a single unit. Okay, you can see the image for the easy understanding. There is given like gene 1, 2 and 3, and there is a P site, which is the promoter site, where usually the RNA polymerase bind. And before and after the promoter sites, you can see certain regions called regulatory sites. So these sites are the sites for interaction by the regulatory molecules, like the specificity. Already we have discussed the specificity factor, activators, or repressors. So these molecules will interact with or near to the site of the promoter that will control the operon. So here, the structure will also give you more idea about operon. There are three genes, and there'll be three proteins produced, and these three proteins, 1, 2, and 3, will be working together, usually working in a single pathway, so that 1, 2, and 3 are required at the same time. That's why they are expressed at the same time. Okay. And about the history, the concept was introduced by Jacob and Monod in 1961, and they discovered such a system in E. coli for the first time for the regulation of lactose metabolism. That's what we study as lac operon. So, what structures are present in an operon? So basically these are the basic structures. That means there should be certain, at least one structural gene. A structural gene means the gene coding for a protein. That means that is the real gene. And there will be regulatory sites also. So there will be structural sites or structural genes and regulatory sites. The regulatory sites include operator site, promoter site, and additional regulatory site. And promoter site, already as we all of us know, that these are the site for binding of the RNA polymerase, which are usually recognized by the sigma factor. And operator is a new site, usually after the promoter site, where a repressor molecule can come and bind, and this repressor molecule usually physically will inhibit the further propagation, for the movement of the RNA polymerase, and thus preventing transcription of the structural genes. And in addition to the promoter operator region, there will be additional regulatory sites. So these sites will usually act as a binding site for the activators. So these activators will enhance the expression of the structural genes. So now we are moving into the basic idea of lacoperon through the description of lacoperon. Lacoperon is actually the gene cluster, and these genes will control the lactose metabolism. Simply the breakdown of lactose into glucose, that means into the simple units. Okay, so these genes are clustered together, and usually the lacoperon will be on. These genes will be expressed only when lactose is present and glucose is absent. That is the most important thing we have to remember. That means lacoperon will be in the active state only when lactose is present and glucose is absent. So how the system maintains this condition? That means how the system will control the expression of genes in the lacoperon when this condition is satisfied. That means when glucose is absent and lactose is present. Okay, so before moving into the mechanism, let's see the structure for again. You can see the structural genes for the lacoperon are Z, Y, and A. This Z codes for beta galactosidase, and Y for permease, and A for transacetylase. And additionally, there will be regulatory sites like operator and promoter sites. And usually this is what's happening. The lactose will come in, and this lactose will be converted into glucose and galactose by the beta galactosidase enzyme. This is what these enzymes will do. And the permease enzyme will act as a transporter for the movement of lactose from the external environment to the internal environment. So both these enzymes are very essential. That means the permease enzyme and the beta galactosidase enzyme for the effective breakdown of lactose into glucose, and the glucose will be further catabolized through glycolysis and energy will be liberated. Then in addition to the structures already described, there will be certain regulatory sites in the lac system, right? So one of the most common regulatory region or a gene usually found in association with lactoperon is the gene for a protein called lac repressor. Right, so this is actually, you can count it as a separate operon, since there is a structural gene called lac-I, and the lac-I will be expressed to produce a protein called lac repressor protein, and this gene is regulated by another promoter site. That means this you can count this as a separate operon. Usually this operon, this small operon, will be located upstream of the actual lac-operon, right? So remember that the lac-I gene is not part of the lac-operon, but instead this is located upstream of the lac-operon, and the lac-I gene will be expressed to produce the mRNA and the repressor protein. And this repressor protein is very crucial for the regulation of lac-operon. Usually this protein has the ability, or which has an affinity towards the operator site of the lac-operon, right? Actually this is a multi subunit protein, and this protein will bind on the operator site. So binding of the operator will prevent movement of RNA polymerase through the structural genes. This is actually a small molecule with around 37 kilodalton, and there will be 2 dimers, and such 2 dimers in 2 units. That means there will be a tetramer. It's a tetramer actually. There will be 4 single units. Right, and usually the interaction occurs at the alpha helix of the protein to the major groove of the DNA. Okay. So this will be the way how this repressor interacts. You can see the operator site, and this repressor specifically interact with the operator, and the polymerase will be, the movement is prevented. So this will be the condition when glucose is present in the medium and lactose is absent, which means there is no need for the expression of the genes for lactose catabolism. So but when lactose is present and glucose has been exhausted in the cell, then in that situation, now we are going to discuss the regulation, how this regulation occurs. So when glucose is absent and lactose is present, what will happen? The lactose, actually not the actual lactose, but instead allolactose. Allolactose is nothing but an isomeric form of lactose. When lactose is present in small concentration in an environment, that there will be an isomerization into allolactose. So this allolactose will act as an inducer. That means allolactose will interact with the repressor protein. So binding of allolactose with the repressor protein will render this repressor inactive. That means there will be a certain conformational change, so that the repressor further cannot interact with the operator, and moreover which also bind on to a repressor which already bound on the repressor, since the interaction site for the repressor and for interaction site towards the operator are different. So the binding of the repressor to the operator will cause release of the repressor even if it is bound to the operator site. So this will make the operator free and allows RNA polymerase to move further, and there will be expression of the genes required for the breakdown of lactose. So you can see the allolactose structure and how this is formed. So allolactose is the actual inducer which causes the activation or the induction of the system. So there are certain other examples also, which they can also act as inducers, like IPTG, isopropyl thiogalactoside, which can also induce lacoperon. That means this IPTG can also bind to the repressor protein and activate the expression of genes. But one of the peculiarity of IPTG is that IPTG is not a substrate for beta galactosidase, right? That means IPTG can induce lactoperon, but IPTG will not be catabolized by the beta galactosidase. So IPTG can act as an effective inducer of lactoperon. So this is the basic regulation of lactoperon, and this kind of regulation usually we have categorized them as the negative regulation of lactoperon. Most of the genes in bacterial system are under negative regulation. So this is a negative regulation of lactoperon. The further regulatory mechanisms like a positive regulation of lactoperon we will discuss in the next videos. Thank you for listening.